Hello guys, I am Shubham from ShubhVed Tutorials. This video is about DNA repair. So before we start, subscribe to our channel and press the bell icon to get notified whenever a new video is out. So let's get started with the video. So in the previous video, we have discussed about replication, all about replication. Now, why repair has to be done? So first of all, we know that the errors have been corrected by the proofreading activity, right? Okay, so proofreading means what? Incorrect nucleotides, if are added during replication, those are removed out. Okay, but sometimes the proofreading activity is escaped. Okay, means replication escapes proofreading and the errors get fixed inside. Okay, errors get fixed. In which cases repair has to be done? First reason is a replication error. Replication errors. Okay, means there are errors during application which incorporate incorrect nucleotides. Okay, so we can say this escapes proofreading. Proofreading activity is escape and application is continued with the incorrect nucleotide. Right? This is the first reason for the DNA repair. Second reason is DNA damage. DNA damage. So DNA damage occurs because of chemical reactions, right, inside the body. Or we can say there are some agents, chemical agents, which bring about damage. Okay. Then we can say radiation, right? So this is caused due to chemical reactions. Then we can say chemical agents. Agent means these are the chemicals only known as mutagens. Mutagens are the chemical substance that bring about mutation. Okay. As well as radiation. Radiations. So this reaction, this bring about DNA damage, right? Means what happens? Main thing is addition of incorrect nucleotide. Addition or deletion of incorrect nucleotides. Got it? Basically, this incorrect nucleotide gets base paired, okay, and then it leads to replication errors or damage in DNA, and that has to be corrected. So that correction is done by these repair processes. Okay, we know that addition or deletion of incorrect nucleotides is known as mutation. Alright, this is known as mutation. So these replication errors and DNA damage leads to mutations in the DNA. So let's see first of all a basic intro DNA mutation. So this first is A is transition. Transition means purine to purine or pyrimidine to pyrimidine. Like A is replaced by G or G is replaced by A. This is purine. Purine to purine. Change. And second, C is replaced by T or T is replaced by C. This is known as pyrimidine to pyrimidine. So when there is change in such type of nucleotides, it is known as transition mutation. Okay, base substitution, right? Base has been changed. Here, this is known as transversion. Transversion. What is transversion? Purine to pyrimidine and pyrimidine to purine. So this is vice versa. Means A can be replaced by G as well as C as well as T, anything. T can be replaced by G, C, A. Okay. So like this, it can be replaced by any nucleotide. In the first case, purine to purine, pyrimidine to pyrimidine, and here B, purine to pyrimidine can occur. Okay. See, now this is the replication error. First round of replication, misincorporation. See, this is misincorporation leads to this is correct replication and this is incorrect because here it is A, right, and here it is G. Here should be T, right? Instead of T, there is G. Now this will go to repair. Okay, this has to be repaired because there is damage in DNA. See, we can see here the structure. This is highlighting the damage. This is known as lesion. Okay, DNA lesion. So these lesions are there means there is a requirement of a repair here. There is incorrect nucleotide, right? So this goes to the repair pathway. The second round replication has been done and here are the incorrect nucleotides. See, means A is complementary T and G complementary is the C, right? So this is a replication error, okay? I will write here, this is about replication error. Now let's see DNA damage. DNA damage occurs because of three reactions. First, A is deamination, means the amino group. This is removed out. Okay, so we can see here removal. Then this forms uracil. Okay, right? This uracil is not found in DNA, but if this reaction occurs, it forms in the DNA. Right? Then deamination here is an example of C. Also adenine, guanine, other nucleotides give rise to deamination. Right? So the pairing is affected. Pairing is affected. C pairs with the, we know that C pairs with G, but U will pair with A. But U is not found in DNA. So this will be unnatural base. We say unnatural base in the DNA. So if this is found, this has to be repaired. Right? Now this B reaction is the depurination. Depurination. What happens in this? The purine ring, whole purine ring is removed. See. So after purine ring is removed, what happens? OH is means sugar is there. Okay, ribose sugar is retained, but nucleotide is removed out. So this is depurination, and the nucleotide will be called apurine, apurinated, right? Apurination. Now this C is the same deamination of methyl cytosine. So this is 5-methyl cytosine. I will write here 5-methyl cytosine. So cytosine on the fifth carbon there is transfer of methyl group, right? See here, we can see methyl group. So how this methyl group is transferred by the enzyme methyl transferase. Okay. Then if this is amino group is removed, it looks like a thiamine. If there are methyl cytosines, then these are regions of hot spots of the mutations. Okay, mutation prone, we can say high chance of mutations are seen. We know T is the natural base. T is found in DNA. Okay, I will write here with a different color. This is a natural base. Natural base means found in DNA. But this uracil is unnatural base, not found in DNA, not found in DNA. So it can be recognized, right? Means it can be recognized by the different enzymes that it is an incorrect nucleotide. Okay, unnatural base. So this has to be removed, right? But in the case of natural base, which is the third case, in the case of methoxytosine, this cannot be recognized because T is found in the DNA. So these are the regions of spontaneous mutations, methylated C. Okay, these are the regions of mutation prone regions, right? So this is about what we have seen here, DNA damage, right? Chemical reactions which give rise to DNA damage. Also deamination, see adenine, deamination of adenine gives rise to hypoxanthine. Hypoxanthine pairs to C, right? Hypoxanthine. And guanine deamination gives rise to xanthine. Which pairs to C? Pairs to C only. So this is natural pairing, but this is unnatural pairing because A has to pair with a T, but instead of that, it paired with C, right? Now after this, there are other reactions too. Alkylation, means addition of alkyl groups, methyl, ethyl, right? So these are the positions where alkylation take place. Then this is the position where deamination take place. And oxidation. See, oxidation is very harmful. There are oxidative species, then oxidizing agents. So when oxidation of this guanine takes place, it results in the formation of oxo-G adducts. See, oxo-G is very harmful. This leads to cancers. Highly mutagenic products are formed by these reactions. Okay, when these reactions take place, highly mutagenic products are formed which lead to cancers. Basically, this oxo-G is the product of oxidation of guanine, right? Then this oxo-G is harmful found in cancers. Okay. Again, this is one type of thymine dimer. So what happens here? Thymine. Two thymines are there. Those got fused to each other. It is a cyclobutane ring here which has formed. So there is fusion. Basically, this is seen in the pyrimidine. But this results due to radiation, ultraviolet, that is UV rays from sunlight, very harmful, right? The radiation causes what this is called? This is thymine. Thymine is pyrimidine. So pyrimidine dimer. Pyrimidine fusion is seen because of this ultraviolet radiation. right? So this occur between thymine dimer. This is common. Also, it can occur between thymine and cytosine, right? Because of UV radiation. Again, these are agents, base analogs. A is about base analogs. What is base analogs? So these are chemicals which resemble to the nucleotides. This resembles to the nucleotide bases, right? See, we can see here bromouracil, 5, 5 bromouracil, ketototamer, but uracil has to be paired with adenine. But this uracil, enoltetotamer, pairs with guanine, right? This is A, base analog. Analog means similar structures to the bases, right? And this B is about intercalating agents. Intercalating agents. See these agents, these chemicals, these are also chemicals, ethidium, then proflavin and acridine orange. So these agents, these chemicals bind to bases, right? Nucleotides. Okay, this gets bound to bases and then this result in the mutations. Basically what this cause, addition or deletion of nucleotides, or we can say bases, because this get bound to the bases, right? These all are the reasons behind the DNA damage, right? First we saw these reactions, deamination, depurination, deamination, then oxidation, alkylation, okay? Then this is because of radiation. Radiation. The radiation causes pyrimidine dimer, okay? Fusion of thymine. Then your base analogs and intercalating agents, right? This all results in the DNA damage, basically mutation. Now there are different types of repair pathways, okay, repair mechanisms we can say, to repair these mistakes, replication errors and the different types of damage we discussed. So that we will discuss from the next video. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section.